In this video, we are going to have a lesson on electron transport chain and how it aids in ATP synthesis in the body. In the previous video, we established the fact that when 1 mole of glucose passes through the glycolytic pathway, produces an equivalent of 2 NADH, and also in the Krebs cycle, produce an equivalent of 3 NADH per 1 molecule of star code A and 1 FAGH2. Now the question is, how is these two molecules, the NADH and the FADH2, useful to the body? The NADH and FADH2 are called electron equivalents. They carry electrons which are used to generate an energy in the mitochondria, which an enzyme called ATP synthase uses to produce or generate or to synthesize ATP in the body by a mechanism we call an oxidative phosphorylation. So in this video, we're going to look at the various components of the electron transport chain system, which aids in ATP synthesis by oxidative phosphorylation, and also how the various components function in the entire complex. The system is embedded in the mitochondria, precisely in the mitochondria membrane. When we take a mitochondria like this is a mitochondria. Mitochondria displays two main membranes. It has an outer membrane and an inner membrane. So here, let's say this side is the cytoplasm of the cell. This is the outer membrane and the inner membrane. Now the space between the outer membrane and the inner membrane are what we call the intermembrane space. So this space is between the outer membrane and the inner membrane of the mitochondria. Generally, the outer membrane is permeable to several ions. For example, this membrane, the outer membrane, is permeable to hydrogen ions and some proteins. So in the normal sense, the content of the cytoplasm or the cytosol and that of the intermembrane space are generally the same, because the membrane, the outer membrane, is permeable to almost the substances in here. So if you have, let's say, a pH of, let's say, 10 in the cytoplasm, it is going to be the same in the intermembrane space, because this membrane is permeable to hydrogen ions, which causes pH balance. Embedded in the inner mitochondrial membrane are several complexes, and these complexes are what we call protein complexes, which helps in the electron transport chain system. Unlike the outer membrane, the inner membrane is not permeable to any ions. So every ion that crosses the membrane of the inner mitochondrial membrane must do so by an active transport mechanism, by which energy is used to propel such substance or an ion across the inner mitochondrial membrane, because it is not permeable to any ion. These four complexes forms the electron transport chain, and these ones are embedded in the inner mitochondrial membrane. There are basically four complexes in the inner mitochondrial membrane that aids in electron transport chain, though each of these complexes have several 1, 2, 3 or more electron carriers in them. Aside the complexes, in the membranes are some other molecules who just help in the transport of the electrons outside the various complexes. The complexes in the transport chain or the inner mitochondrial membrane are complex 1, complex 2, complex 3, and complex 4. The transport of the electrons across the various complexes are aided by the general or the standard reduction potentials of the various complexes. So the reduction potentials of the complex 1 to 2 to 3 and 4, basically they differ. So the differences in the reduction potentials causes the ability of the electrons to be transported from the electron carriers or the electron equivalents to the final electron or the final protein complex in the inner mitochondrial membrane. Now, having here, aside the complex 1, complex 2, complex 3, and complex 4, we have other substances we said earlier that aid in electron transport. The first one we talk about here is the coenzyme Q. Coenzyme Q, which is also called ubiquinone. Ubiquinone as in ONE, a ketone functional group. Ubiquinone. This complex is embedded in the bilayer or the lipid bilayer of the inner mitochondrial membrane. Another one is cytochrome C. The cytochrome C complex is also embedded or is outside the inner mitochondrial membrane, so it's within or on the upper part of the bilayer, which shows to the intermembrane space. So the coenzyme Q is soluble in lipids, so it is embedded in the inner mitochondrial membrane, but the cytochrome C is insoluble in the lipid, therefore it is outside the inner mitochondrial membrane. So let's see how these various complexes aid in electron transport in the electron transport chain. Basically, the NADH is generated in glycolysis. It is generated in conversion of pyruvate to acetyl-CoA in anaerobic respiration, and also severally in the tricarboxylic acid or the Krebs cycle or the citric acid cycle. The FADH2 is produced in the Krebs cycle precisely. So these two, the FADH2 and the NADH, are electron carriers that carries electron and give it the various complexes and transport them to the final electron acceptor in which is embedded in the mitochondrial matrix called oxygen. So basically, in mammalian electron transport chain, the final electron acceptor here is oxygen, and that oxygen is get reduced to water at the end of the reaction. The final or the initial electron donor in the electron question is the NADH or the FADH2. The NADH is produced in the glycolysis and it is shuttled to the mitochondrial matrix by the phosphoglycerol shuttle. Then the ones which are produced in the Krebs cycle directly enter the transport chain system. Basically, the NADH gives the electron it carries. Here it donates more of a hydride ion. So the hydride ion carries 2 electrons and 1 hydrogen. So the hydrogen comes in the matrix, while the electrons goes in the first complex. Bear in mind, the NADH gives its electron to the first complex. Then the complex shuttles the electron to the other complexes till it gets to the oxygen. The FADH2 gives its electrons to the complex 2 initially. The FADH2 gives its electron to complex 2 initially before it passes on to coenzyme Q or the ubiquinone to the other complexes. Now, due to the oxidative reduction actions of the various complexes, this gives them different names. So let's see how the various complexes are named. The complex 1 gives or takes electron from the NADH and oxidizes it and gives it to coenzyme Q and reduces it. So the name of the complex 1 is called NADH dehydrogenase, or it can also be termed NADH coenzyme Q oxidoreductase. The complex 2 takes electron from the FADH, and these ones are produced in the Krebs cycle at conversion of succinate to fumarate. So the complex 2 is termed succinate dehydrogenase or succinate coenzyme Q oxidoreductase. The complex 3 also receives electron from coenzyme Q and gives it to cytochrome C. So the complex 3 is termed coenzyme Q dehydrogenase or coenzyme Q cytochrome C oxidoreductase. Then the complex 4 is called cytochrome C oxidase, as simple as because it takes oxygen or electron from the cytochrome C and oxidizes it. Now let's see how the various complexes aid in electron transport in the electron transport chain. Now here, these are the various 4 complexes: complex 1, complex 2, complex 3, complex 4, and this the enzyme ATP synthase, which takes the proton or the energy generated to produce ATP by oxidative phosphorylation. Here is the intermembrane space. Down here is the mitochondrial matrix. When NADH gives its electron to complex 1, the complex 1 has several electron carriers embedded in them. We have in the iron sulfur complex, which is very mobile, which catalyzes or transfers electron to the other complexes in the inner mitochondrial membrane. When electrons are given to complex 1, the complex becomes excited, and this excitation causes it to pump protons from the matrix to the intermembrane space. When it receives electron, it causes it to be excited, and the excitation is used that excitation energy to pump protons from the matrix to the intermembrane space. By pumping, it loses the electron to the next electron transporter in here, which is coenzyme Q, ubiquinone, or also called vitamin K2. So complex 1 gives its electron to coenzyme Q upon the pumping of proton from the matrix to the intermembrane space. Now the coenzyme Q is very mobile in the lipid bilayer, so it flows in there and gives the electron to complex 3. Now the complex 3 has several electron carriers too, which transport the electrons in its complex, then gives it out to cytochrome C. As we established earlier, the cytochrome C is not soluble in the bilayer, so it is found outside or in the intermembrane space. So the complex 3 gives the electron to the cytochrome C, and the cytochrome C also gives the electron to complex 4. Then the complex 4 gives the electron to oxygen to reduce it to water. The FADH2 does not give its electron to complex 1, but rather it gives it to complex 2. So when complex 1 gains an electron, it becomes energized and pumps electron or protons from the matrix to the outside. When complex 3 receives electron, it becomes energized and uses that energy to pump protons from the matrix to the intermembrane space. I think per the gaining of 1 electron, it pumps 2 protons out. When complex 4 receives an electron, it pumps protons out of the matrix to the intermembrane space. FADH2 gives the electron to the complex 2, but the energy it gained is not enough for it to pump the protons out. So among the 4 complexes, it is only 3 complexes that pumps proton out of the matrix to the intermembrane space. So the FADH2 gives the electron to complex 2, but it can't pump protons out of the matrix because it has less energy, but gives the electron to coenzyme Q or ubiquinone. Then the ubiquinone, which gets reduced to ubiquinol, gives the carried electron to complex 3, and complex 3 becomes energized and pumps proton out of the matrix to the intermembrane space. The same routine, it gives it out to cytochrome C, then cytochrome C to coenzyme or complex 4 also pumps the proton from the matrix to this one and gives the electron to final electron acceptor in the chain called oxygen, and this oxygen gets reduced to water. So by the flow of electrons from the various complexes, we are going to have partial charges, more negative charges in the inner, and the more positive charges in the outer. So the outer ones are because of the increased concentration of the hydrogen ions. Now these different charges create an energy we call an electrochemical gradient or a protomotive force or a proton gradient. This energy is what is used or it serves as an electric current, which the ATP synthase uses that current to add a phosphate group to ADP to produce ATP. This mechanism of ATP synthesis by which the flow of protons or the use of electrochemical gradient is done by the ATP synthase to produce ATP from ADP and phosphate is called oxidative phosphorylation. Bear in mind, we said earlier that in substrate level phosphorylation, a phosphate group is taken from a substrate and added to ADP to form ATP. Unlike here, the electrochemical gradient created as a result of inflow or the outflow of the hydrogen ions are used by the ATP synthase enzyme, which is embedded in the mitochondrial membrane to produce ATP in the mitochondrial matrix. So that's how basically electron transport chain occurs It has four basic complexes. The final electron acceptor here is oxygen. The initial electron donor here is either NADH, FADH2. The NADH gives its protoelectrons to complex 1, and FADH2 gives its electron to complex 2. The coenzyme Q serves as an electron receiver from both complex 1 and complex 2. ATP synthase uses the proton gradient, protomotive force of electrochemical gradient to produce ATP upon the flow of the hydrogen ions back to mitochondrial matrix to disrupt the gradient. Thanks for watching. Don't forget to subscribe. In the next video, we're going to look at beta oxidation of fatty acids and how they also aid in the production of acetyl CoA. We'll look at fatty acid synthesis and various topics. Thanks for watching. Don't forget to subscribe. Thank you.